thousands of improvements and discoveries that happened between the discovery of microscopes and then the century of biology, where we've really started to understand so much more about microbiology. But I wouldn't quite be right unless we went through a summary of the prominent discoveries made in the past 300 years. First of all, microscopy really launched the field of microbiology. Without microscopes, the awareness of this widespread distribution of microorganisms was vague. Then there's the rise of the scientific method. The rise of the scientific method helped us understand what caused what, and then the development of medical microbiology. So here's a quick tour through some of those aspects. People have been aware for a long time of the effects that microbial growth has. For example, spoilage of food, the passage of disease, and decomposition. However, microbes are far too small to be seen even with hand lenses. And so their widespread distribution wasn't really possible until development of the first microscope. At this point, the study of diseases like the Black Plague and smallpox were cloaked in mystery, even though some people understood that they were caused by some sort of transmissible element. There still remained a lot of superstition, and the concept of spontaneous generation was holding the lead. Now, spontaneous generation means life from non-life. For example, flies arise directly from meat, because of course maggots couldn't be seen on the meat developing into flies. So that brings us to the work of Robert Hooke. He was an English naturalist and an architect. And he was the first to really see microorganisms and coin the word cell to describe what he saw. He took a look at almost everything, household items to plants and tree bark. In fact, he drew cellular structures in tree bark using his simple magnifying lens. He looked at spots of mold on his sheepskin book cover. These are what we can see in his images here. The one in the circle is what he drew in his notebook. And here you can see a slide of the same type of fungi. It's the work of people like Robert Hooke that paved the way for Antoine van Leeuwenhoek. In 1684, about 20 years later, he's the one that gets credit for designing the first true microscope. He was a Dutch fabric merchant, and he wasn't really interested in biology at all. He was interested in making this microscope to look at thread counts. So between customers, he would spend time in the back of his store working on his microscope lenses to make the first true microscope. You can see images of this prototype over here. He had a specimen holder and a focus knob and all kinds of the components that we see on today's microscopes. He became more and more curious, looking at things from plaque that he found off of his teeth to water samples. He noticed that these water samples were literally alive, and he named the little things in them animalcules. His descriptions of these animalcules, which are really known today as protozoans, were so accurate that he's considered to be the father of microbiology. So the key to the study of microorganisms was the development of the microscope. The earliest record of those was Robert Hooke. And then Leeuwenhoek created the first microscope, single lens microscope, and became known as the father of bacteriology as well as protozoology. He revealed microbes as discrete living entities. All these discoveries allowed the awareness of widespread. All of these discoveries allowed a new awareness of how widespread the distribution of microorganisms really was. More recently, we've had some discoveries that had huge impacts on our understanding of microbiology. In the 1970s, for example, we discovered restriction enzymes. Restriction enzymes are little molecular scissors that are found inside prokaryotes or bacteria that can literally chop up DNA in specific ways. This allows the insertion of one gene into a totally different genome. It's because of restriction enzymes that we were able to isolate the gene for human insulin and insert it into certain bacteria to allow them to produce infinite amounts of human insulin. Another critical invention was the invention of the PCR technique. PCR is polymerase chain reaction. It came about in the 1980s, and it was a breakthrough in our ability to detect tiny amounts of DNA and amplify them into sufficient quantities for studying. For example, what if you found a tiny bit of DNA at a crime scene? It wasn't enough to run a DNA fingerprint on. Because of PCR, we could take that tiny fragment of DNA and multiply it millions of times in order to create an unlimited amount for continued study. In the 1980s, 90s, and 2000s, we've been particularly interested in biofilms and their influence in infectious diseases. Biofilms are accumulations of bacteria and other microbes on the surfaces of things like your teeth or a shower curtain. Some of these biofilms, for example on our teeth, might be responsible for infections that are tough to conquer. I just learned recently when I went to see my dental hygienist for a teeth cleaning that these biofilms can also give us a prediction of some of the issues that people are having internally. For example, there are certain bacteria species that are found in biofilms on our teeth that are also responsible for some issues in heart disease. Interesting, huh? Also, ear infections or infections in the prostate could be predicted by these biofilms. In the 2000s, we found out that there were many other RNA pieces, and these RNAs, not just messenger RNA or tRNAs that we were already familiar with, but other RNAs that are involved in regulating what happens inside the cell. It's not just important for the correct scientific assumptions, but there are important practical uses as well. It's led to new approaches in how diseases are treated, because these small RNAs actually influence how genes are expressed. You may have heard of them in the sense of epigenomics. Establishment of the scientific method allowed us to more methodically understand and analyze things in science. Early scientists tended to explain natural phenomena as a mixture of belief and superstition and then just simple argument. But during the 1600s, true scientific thinking developed. From that, we had development of the scientific method. This is the place in which we formulate a hypothesis and use usually a deductive approach, but it could be an inductive approach, and apply this scientific method where we do experiments based on the hypothesis, analysis of results, and testing that leads us to conclusions. These conclusions either will refute or support the hypothesis. Now, the key with any of these hypotheses then is that they have to be falsifiable. That means they have to be refutable. So we can only use scientific explanations. An example here is I can't say, I wonder why my car won't start. There's the question. The hypothesis, I bet that it won't start because I'm not supposed to make it to work today. Now, I can't falsify that hypothesis. There's no amount of testing or analysis that would possibly allow me to falsify that. It's just my own belief about why I think my car might not start. However, if I apply the scientific approach, I would go through and I would say, I hypothesize that I'm out of gas. So I can test that hypothesis. I can either show I have gas or I don't have gas. I hypothesize that the starter's broken. And after a number of hypotheses and refuting or supporting those hypotheses, I'll be able to establish what the most likely explanation is. Now, of course, science changes all the time, and there's some mystery in the fact that we use the word theory to describe the things we understand the best. Basically, in science, theory is virtually a fact. The theory of gravity, for example. We pretty much know that gravity exists. Now, later on, we might call it something different and explain it in some other means, but for now, that's our belief of what gravity is. That's why we use the word theory instead of fact, because we're always open to an alternative solution. We used to think that the world was flat, and it wasn't until we developed other technologies. Float around on the ocean, we found out that the Earth indeed was spherical. But either way, establishing a scientific method allowed us a much more methodical way to describe these natural phenomena. Now, the development of medical microbiology came about because early experiments on the sources of microorganisms led to the profound realization that microbes are absolutely everywhere, not only in the air and in the dust, but on the entire surface of the earth, in its waters, and all objects are inhabited by microbes. This discovery led to immediate applications in medicine, and thus the need for medical microbiology. So we'll begin with the discovery of spores and sterilization. We're all familiar with the name Louis Pasteur. He worked on pasteurization. He did a lot of work with infusions around the mid-1800s. And he showed that air dust, or dust that's in the air, could be a source of microbes. Then John Tyndall showed evidence that some microbes have very high heat resistance and are difficult to destroy because in his solutions, even after heat application, some microbes were thriving. And then it was the work of Ferdinand Cohen who discovered that there were actually endospores that were heat resistant. So that tied together the idea of Louis Pasteur's air dust being a source of microbes, John Tyndall showing that regular microbes have very high heat resistance and can't be destroyed, perhaps because they were spores found in the air. These are the ones that Ferdinand Cohen discovered. Once we understood that these spores were heat resistant and could not be completely eliminated, it was necessary to develop the aseptic technique. Physicians and scientists also began to suspect that microorganisms could cause disease. The way they came about this assumption is kind of nasty. Someone made the observation that women who gave birth in hospital were more highly likely to end up with infection than women that gave birth at home. They further discovered that women in the maternity ward became infected after physicians came directly from the autopsy ward. Yuck. So development of an aseptic technique was absolutely necessary. In fact, you'd be surprised that as recently ago as the late 1800s, surgeons wore street clothes in the operating room and had little idea that hand washing was even important. It's all of this that drove Joseph Lister to introduce the aseptic technique. Let's take a moment to look a little closer at the work of Louis Pasteur and Robert Koch. Louis Pasteur, as you know, is responsible for pasteurization. He gave us insights into the microbial role in wine and beer making. And pasteurization is used on all of our dairy products these days, which is unfortunate. We can't readily find unpasteurized dairy products with their beneficial living organisms. But Pasteur also showed human disease could arise from infections. And his studies, supported by the work of others, became known as the germ theory of disease. The germ theory of disease is that microorganisms can be the cause of disease, literally that germs cause disease. Now, Robert Koch, one of his colleagues, came up with a series of proofs known as the Koch postulates. They verified the germ theory and could establish whether an organism was pathogenic or which disease it caused. For example, Koch discovered that anthrax was caused by a microorganism called Bacillus anthracis. He realized that the study of microbes will require separating them and culturing them. He developed most of the techniques that we're going to learn in the next chapter, along with the techniques of sterilization that were provided by Lister. That should be an exciting chapter. But before we go there, we'll spend some time in the next section learning how we name, classify, and identify microorganisms.